Hello everyone, I'm Dr. Monica. Today in this video, let's understand the pathogenesis of periodontal disease in a simple way. So what is pathogenesis? So pathogenesis is derived from Greek terms pathos, which means suffering, and genesis, meaning generation or creation. According to Merriam-Webster's Collier Dictionary, pathogenesis is defined as the origination and development of a disease. In simple terms, pathogenesis refers to how a disease begins and develops. So, plaque bacteria clearly initiates and maintains the inflammatory response in the gingival tissues. But the major determinant of the periodontal disease severity lies in how the body's own immune inflammatory response is towards the bacterial challenge. So when we look at the periodontal pathogenesis, the periodontal pocket, it acts as a very favorable environment for the survival of the bacteria. So many of the unique features of periodontitis, it is derived from the anatomy of the periodontium itself. That is, the tooth provides a hard, non-shedding surface for the bacterial colonization, and it is partly embedded within the connective tissue. It crosses an epithelial surface and it is also exposed to the external environment. So this creates a unique scenario where the bacteria, it remains outside the body on the tooth surface, but the inflammatory response it triggers occur within the body. So plaque-induced gingivitis, it is the most common form of periodontal disease. It begins with plaque at the gingival margin and it can spread throughout the remaining gingival tissue. So we can see prevalence increases in children peaking at puberty. So in inflammatory response in older adults, it tends to be more pronounced even with smaller amount of plaque, which highlights the role of age-related changes in host response. In plaque-induced gingivitis, we see clinical signs of inflammation such as contour, color, and consistency, but the periodontium exhibits no loss of periodontal attachment or bone loss. The common clinical features include erythema, edema, bleeding, sensitivity, tenderness, and gingival enlargement. Chronic periodontitis originates as plaque-induced gingivitis. It is a reversible condition and it may progress into chronic periodontitis if left untreated. It has various clinical features. For example, there is alteration in color, texture, and size of marginal gingiva. There is bleeding on probing from the gingival pocket. There is also increased pocket depth with loss of probing attachment level. We can see loss of alveolar bone and recession of the gingival margin. There is involvement of root furcation. Also, there is increased tooth mobility, and we can see pathological tooth migration. And in advanced cases, there is tooth exfoliation. Now talking about plaque-induced gingivitis on a reduced periodontium, it occurs in areas with previous attachment and bone loss without ongoing tissue destruction. It presents clinically like the regular gingivitis except for the presence of pre-existing attachment and bone loss. So coming to the histopathology of gingivitis as given by Page and Schroeder 1976. So gingivitis, it is divided into four stages. The first one starts from initial lesion, followed by early lesion, then established lesion, which finally progresses to advanced lesion. The initial lesion, clinically it is represented by the healthy gingiva. In this we can see slight increase in the vascular permeability and vasodilation, and there is a flow of GCF which begins here, and we can see migration of neutrophils through the connective tissue and junctional epithelium into the gingival sulcus. Next stage is the early lesion represented clinically by gingivitis. There is increased gingival flow here with infiltration of neutrophils and lymphocytes, and there is beginning of collagen destruction along with fibroblast degeneration. We can see epithelial proliferation into the connective tissue. Now early lesion progresses to established lesion, which is clinically represented by chronic gingivitis. Here we can see dense inflammatory infiltrate represented by plasma cells, lymphocytes, and neutrophils. MMP release from neutrophils and pocket epithelium is formed filled with neutrophils, and we can see significant collagen loss in this lesion. Last stage is the advanced lesion, which is clinically the periodontitis. Here there is apical migration of junctional epithelium and predominantly neutrophils are seen in the pocket with massive collagen breakdown, and we can see osteoclastic bone resorption. Now let's talk about the junctional epithelium and its role in the pathogenesis of periodontal disease due to its unique structure. It has fewer desmosomes, making it intrinsically a leaky barrier. This leaky nature allows immune cells like neutrophils and macrophages to pass through from the gingival tissue into the sulcus to fight the bacteria. Also, this pathway allows the bacterial products and the antigens to enter the tissue, which can lead to inflammation and disease progression. So coming to the pathogenesis of periodontal pocket, the initial lesion in the development of periodontitis is the inflammation of the gingiva in response to a bacterial challenge. Now the pocket formation, it begins with the inflammatory changes in the connective tissue wall of the gingival sulcus. The inflammatory exudate leads to degeneration of the surrounding connective tissue, including the gingival fibers. Just below the junctional epithelium, collagen fibers, they will be destroyed and replaced by inflammatory cells and edema. The loss of collagen, it occurs through two main mechanisms. One is by the release of MMPs and the second one is by the phagocytosis of collagen fibers by fibroblasts. Now the apical cells of the junctional epithelium, they begin to proliferate and migrate along the root surface while the coronal portion detaches from the tooth surface. So inflammation causes the PMNs to invade the coronal junctional epithelium, and when PMNs make up about 60% of the tissue, it loses cohesion and detaches from the tooth, causing the sulcus bottom to move apically. The oral sulcular epithelium then gradually lines more of the sulcus. Early pocket deepening occurs either between the junctional epithelium and tooth or within the junctional epithelium itself. So there is ulceration of the pocket epithelium and finally establishment of periodontal pocket. So coming to the histopathology of lateral wall of pocket, there is proliferative and degenerative changes in the epithelium with epithelial buds or interlacing cords extending from the lateral wall into the inflamed connective tissue or going beyond the junctional epithelium. So these projections also show leukocytic infiltration and edema. There is also vacuolar degeneration along with vesicle formation, and the progressive degeneration and necrosis leads to ulceration of the lateral wall and exposing the underlying inflamed connective tissue and leading to pus formation. There is also acute inflammation overlaying the chronic changes. So let's understand the transition from gingivitis to periodontitis. So gingivitis always precedes periodontitis, but not all gingivitis progresses to periodontitis. So plaque accumulation is necessary, but it is not sufficient alone because a susceptible host is also required for periodontal disease progression. We can see increase in B cells from health towards gingivitis, then towards periodontitis. So B cell to T cell ratio also increases with disease severity, and this states that high B cell levels in active lesions suggest B cell activation contributing to disease progression. So inflammatory responses in the periodontium, they are triggered by various microbial virulence factors such as lipopolysaccharide, bacterial enzymes and noxious products, proteases, microbial invasion of tissues, fimbriae and extracellular bacterial DNA, and other microbial components which we will understand one by one. Talking about lipopolysaccharides, they are found in gram-negative bacteria. They are recognized by toll-like receptor number 4 and it triggers host immune and inflammatory response. So bacterial enzymes and noxious products, they directly cause the tissue damage. Also, they aid in nutrient supply via tissue destruction and influence the cytokine secretion, and they potentiate inflammatory responses. Talking about proteases, gingipains, they play a key role in periodontal pathogenesis by degrading the host proteins such as collagen, elastin, and fibronectin. They also disrupt host immune defenses by inactivating TNF alpha and activating protease activator receptors, which leads to increased cytokine secretion. So next is microbial invasion by P. gingivalis, A. actinomycetem comitans, and F. nucleatum. They invade epithelium and connective tissues and persist intracellularly, act as reservoirs for reinfection. So next is fimbriae, which help attach to pellicle, and extracellular bacterial DNA contributes to biofilm structure and virulence. So let's understand the host-derived inflammatory mediators. So whenever there is presence of dental bacterial plaque, it triggers the host inflammatory response to release inflammatory mediators such as cytokines, prostaglandins, and matrix metalloproteinases. So main cytokines involved in periodontal pathogenesis include interleukin 1 beta, which induces mediators and activates immunity, interleukin 1 alpha, which signals from necrotic cells, also known as alarmin, interleukin 1 Ra. They are anti-inflammatory and they block the interleukin 1 beta. Interleukin 18, they activate neutrophils and they are T cell chemoattractant. TNF alpha, they increases the neutrophils and they induce apoptosis. Interleukin 6, they stimulates osteoclasts and they are also elevated in periodontal disease. Now prostaglandins, they are produced by macrophages and fibroblasts. They induce the MMPs, stimulate bone resorption. Now coming to MMPs, MMP1, MMP8 and MMP9, they degrade the type 1 collagen. They are secreted by neutrophils, fibroblasts and osteoclasts, and they contribute to periodontal tissue destruction. So all these inflammatory mediators, they lead to collagen breakdown and ultimately bone resorption and finally periodontal attachment loss. Now talking about the immune responses in periodontal pathogenesis, they are of two types. First one is innate immunity. This is the inherited immediate response with limited specificity and it does not improve with repeated exposure. Second one is adaptive immunity. It is slower but highly specific and it involves T cells, B cells and antigen presenting cells, and it improves with exposure and it provides memory. Now talking about the innate immunity, we have saliva which provides antimicrobial action, and then epithelial tissues, they act as barrier against the bacteria. We have gingival clavicular fluid which brings the immune components into the sulcus. Then the immune system, it detects the pathogens and activates the innate responses. The neutrophils, they form defense barrier in the junctional epithelium, protecting tissue from the subgingival plaque. So in the adaptive immunity, we have antigen presenting cells. These are dendritic cells, macrophages and B cells. They detect and take up the microorganisms and then interact with T cells to present the antigen. T cells, they are the predominant phenotype in the stable periodontal lesion. Any alteration in the balance of effector T cell subsets within the CD4 population may lead to progression toward a destructive B cell dominated lesion. The antibodies to periodontal pathogens are primarily the IgG with few IgM or IgA types. So there exists a concept of host susceptibility, which states that the risk factors for periodontal disease such as smoking, systemic disease, nutritional deficiency, psychological stress, etc., and the host response, that is individuals with hyperinflammatory phenotype, that is they exhibit an exaggerated immune response. All these, both the factors that make the person more susceptible to periodontitis. Now let's understand the molecular aspects of pathogenesis of periodontitis. Microbial products and extracellular ATP and DNA, they activate the inflammasome, which triggers the release of pro-inflammatory cytokines and which further activates the PMNs. The immune activation, it leads to ROS production and neutrophil migration. Mediators such as histamine C3A and C5A, they increase the vascular permeability, which allows the serum leakage from capillaries into the tissue. Keratinocytes, they serve as physical barriers and respond to pathogens by releasing pro-inflammatory cytokines and defensins to kill microbes. Dendritic cells, they capture and present the antigens and express co-stimulatory molecules and cytokines and further activate B and T cells. So when the dendritic cell homeostasis is disrupted, so it can lead to immune dysregulation and which contributes to destructive inflammation. Natural killer cells, they are cytotoxic lymphocytes that kill target cells without prior sensitizations via direct contact, and cytokines such as interferon gamma. They act innately and rapidly, distinguishing self from foreign antigens. Interferon gamma inhibits T helper 2 activity and links NK cells to periodontal inflammation. So coming to T and B cell subsets, Th17 cells, they are elevated in the gingiva of periodontitis patients, contributing to inflammation, whereas the Th2 cells, they support humoral immunity by producing cytokines and promoting the IgE and IgG1 switching and mucosal defense. So aberrant microRNA expression, they disrupt both the innate and the adaptive immunity, causing ineffective microbial defense or excessive inflammation, and it can lead to the pathogenesis of periodontal disease. Linking pathogenesis to clinical signs of disease, increased neutrophil infiltration leads to collagen depleted zones, prompting epithelial proliferation to maintain the tissue integrity. Epithelial cells, they produce defensins including the HBD1 and HBD2 in the gingival epithelium, which play a critical role in shaping the host pathogen interactions and protecting the epithelial barrier. Talking about alveolar bone resorption, it is body's protective response to prevent bacterial invasion of bone but can cause tooth mobility and eventual tooth loss. Inflammatory mediators, they act within the 2.5 mm of alveolar bone, beyond which resorption stops. Osteoclasts, they are activated by pro-inflammatory cytokines and other factors, leading to alveolar bone retreat from the inflammation site. So let's understand the role of receptor activator of nuclear factor kappa B ligand or osteoprotegrin in the periodontal disease pathogenesis. So OPG, it acts as a decoy receptor by binding to rankle and preventing it from activating the osteoclast and reducing the bone resorption. In active periodontal breakdown, rankle levels are increased while OPG levels are decreased, creating an imbalance that promotes bone loss. Consequently, the rankle or OPG ratio in the GCF is higher in periodontitis than the healthy ones, indicating the ongoing bone destruction. If you have liked the video, please like, share and subscribe. Thank you.